Recording in progress.
I wanted to diverge a little bit from presenting a whole bunch of slides and actually dive into a journal article. And I found a journal article that I found very intriguing and interesting that I think will help with a little bit of a description of the overall concepts that I've presented before. A lot of the things contained in this journal article are very complicated. And so I wanted to review it, show some of the diagrams, show some of the patients that are involved in this presentation, and overall just kind of spell out what is being described in this journal article. So at the very top, the journal article is the Oligogenic Origin of Differences of Sex Development in Humans. It was published in the International Journal of Molecular Sciences in 2020. And the idea is that this is a review. This is a collection of a lot of journal articles that were previously published and gathered together to create a description of what's happening among all these journal articles to make a cohesive collection of documents and a description that helps to further advance medical education and science. So that is basically what this is. It's a review. It's a collection of all these journal articles and abstracts and case studies that help to move these projects and understanding along. With this description, down here at the very beginning with the introduction, I love this very first sentence that they use right down here. It says, sex development is a very complex biological event that requires the concerted collaboration of a large network of genes in a spatial and temporal correct fashion. If you've reviewed my previous videos, you've seen my reviews of the sexes, the definition of biologic sex, and how it's very complicated and difficult to get a clear picture or a very defined definition of male versus female because of the amount of variations that we see. It's not just XY, it's not just XX, it's not just the SRY gene, it's not just SOX9, it's not just external structures, it's not just internal structures, it's a collection of characteristics and their variations that lead to sperm or egg development. The next part of this paragraph is also very important in describing these individuals with differences with sexual disorders, differences of sexual development, and that we've only described approximately 50% of these cases. If you were to divide out each one of these cases or these large groups, you can point to the chromosomal differences and say, yes, this person's Klinefelters or this person's Turners. That doesn't describe or capture every single individual, however. When I was in residency and I picked up my Harrison's Guide to Internal Medicine, there's a list of about 56 different conditions that are described in differences of sexual development. And each one of them has molecular genetics that are associated with each one of them. And many of them have several. There are many genes that are, for example, listed right here, congenital hypogonadotropic hypogonadism. They describe more than 25 positive genes. So down here, CHH is now considered to explain around 50% of the cases. So even though we have an idea of about 25 different causative genes, there's still a number of them we don't fully capture or grasp. Mm. They also mention here that this is not the only medical condition that appears to have multiple genes influencing the phenotype that happens to individuals, and they use hypothyroidism as another one, using digenic, which is two genes, or polygenic, which is multiple. As we go into the second page, you can see that they move on to this topic of monogenic, the individuals where there's just one gene that influences the entire phenotype. And this is not just the chromosomes of the chromosomal sex, but they also describe that there's disorders of sexual development based off of the chromosomes. So if you were to categorize this, you could say a chromosomal sex or chromosomal abnormalities, and you could say 46XX or 46XY in terms of these differences of sexual development. And all they're doing in here is just creating a way to categorize these differences and variations. What would be expected with an XX individual, of course, is to develop as a female. Of course, there are variations on that theme that are caused through various mutations that doesn't always become the case. Or there's 46XY individuals who have variations of genes that lead them to not develop as a male. So it's one of those where we're trying to categorize and simplify how we look at each one of these genes in these different situations. As you go down to the next set, you can see a map of a whole bunch of genes listed. This is not all of them. This is only a small collection of all the monogenic genes that they're mentioning here. Down farther, they mention digenic and combined and oligogenic, so multiple genes influencing all of this. And they use an example of an individual with an androgen receptor defect that also happened to be Klinefelter's. So this individual was born with female anatomy. So it becomes much more complicated when you have quote unquote multiple hits to various genes. And that's what we're seeing. It's not just one gene anymore. It's not just the chromosomes anymore. It's actually multiple genes where if one of them is partially influenced and another one's partially influenced, together they may act as one overall gene that has been affected. And it's not even just, two, it could be two or three, maybe four. Um, all of a sudden, this idea of sex development becomes incredibly more complicated. And this is going down each one of these patterns here. Again, just what we're finding here is that with the advances of technology for gene sequencing, that they can create these panels of expected genes for differences in sexual development. But what happens is as soon as you create this panel, the advances that we're making are so rapid that that panel becomes obsolete very quickly. And so it actually makes more sense now to do these whole genome sequences. Take a look at the entire structure, the entire chromosomes, to get a gauge of what's happening with this. However, there are caveats to that because which candidate gene are you looking for? Now that you've captured the whole entire thing, there are variations that may or may not have an influence, and now trying to decipher which one is it becomes much more of a challenge. That's aided by a lot of computer technology and algorithms, but again, we have to get to that development point where we can capture not just the monogenic ones, but the multiple genes that are causing problems. Right here is a table of, if you look over to the right, where it says references, they went through each one of these journal articles and they basically said, we're going to go back and we're going to take a look at each of the individuals and what gene sequences, which genes, not just the first gene that was affected, but what were the other complementary genes that were interrupted as a consequence of this. Some of the ones that are probably the ones that stand out to me the most is the androgen receptor by the acronym AR. You can see with this one, the SOX9 and POR were also affected amidst all this. SOX9 is probably one of the more important genes in male development. So that one becomes very straightforward. You can see that on a pathway. But each one of these genes, WT1 and GATA4, very much have a role in male or female development. Again, here's androgen receptor, there's the SRY gene. It becomes much more complicated now that we've added multiple genes into this sequences. And this table goes down for a little bit farther. Um, In this page, they take each one of these journal articles and they list out really what they're identifying. So the top paragraph says we had 37 candidate genes and 21 CHH genes in these individuals. We found several to carry gene variations in more than one candidate gene. Basically, each one of these paragraphs describes these situations where it's not just one gene, it's multiple genes that are in the influence of this. So this goes back to the references down at the very end and says, okay, here's the journal article. Here's what they found in this journal article as an overall summary. and that continues down for a little bit. For those who have seen my previous videos, this slide might actually be familiar to you. This takes through the gestational development of sex, starting with sex determination with the gonadal ridge, where both structures, male and female, the Mullerian and the Wolffian ducts, are present, and how we go from that structure to male structures and female structures. And you can see that there's a lot of connection and relationship of these genes that work on each other. Some of them are positive influences, some of them are negative. As I mentioned on that previous table, here's that SRY gene that's influencing SOX9. Down here is the androgen receptor, the anti-Mullerian hormone. There's WT1. So again, what we're seeing here is a small collection of a lot of the genes that have already been mentioned. This does not include all of them. There are multiple genes and pathways in the midst of all this that can be listed and categorized, but it becomes very complex to try to picture this on a on a sheet of paper. It would take a large map to see all these sequences and genes and interactions. And as you can see at the top of there, there's a whole bunch of candidate genes, and down at the bottom, there's other candidate genes. There's a lot of genes that make us who we are. Again, really emphasizing the fact that it's not just the X or the Y chromosomes, it's that collection of genes that leads us to be who we are. Oops, now I'm going way too fast. So the last part of this journal article, they take a gene sequence, the MAML3 gene, and they basically throw it into a whole collection. So there's the MAML3, and they show all the interactions of MAL3 in the body. Now, as you can see, this tree and interaction is incredibly complex. And as a consequence of that, if you affect one gene in one protein, it has a cascade effect. If you have multiple genes that are somehow influenced and affected by this, that cascade effect becomes more and more pronounced. The last part of this talks about some of the pitfalls. Again, going back to these gene panels where not all the genes may be included in a gene panel. And we have to be careful with that because there are people that do get tested where things come back negative. Well, it certainly could be a false negative. Maybe that gene panel did not include the individual's gene that has been affected, or the multiple genes, or the variations haven't been quite discovered yet. Whereas if we do a whole genome sequencing, where we look at the whole entire body, then we run into the problem of, well, maybe there's too much information. We gather everything, and how do we derive it down into the simplest form and say, this is the gene that's the causative effect of this? So overall, in summary, I'm going to go back up to the abstract. This review in the Journal of Molecular Sciences, basically what it's doing is it's gathering a whole bunch of studies that looked at individuals with disorders of differences of sexual development. And the biggest key point is here. The genetic cause of less than 50% of individuals has been solved. We don't account for more than 50% or roughly 50% of individuals. The other key point behind this is that the overall development, our development as human beings, is incredibly complex. And the more that we dive into our genetics, the more we realize that it's not just one thing. It's a collection of things. It's not just one gene all the time. It's multiple genes that are working in concert to each other. That there's variations in genes that lead to problems down the line in terms of our development. So overall, a very fascinating study. This was performed in 2020, and I will have a wonderful time actually looking through the references and pulling out some more of these journal articles. Thank you for your time.